Our next speaker is Professor Michael Brown. Professor Brown is originally trained as a clinical and laboratory immunologist. And after a period of laboratory and translational research in cancer immunotherapy at St Jude's Children's Research Hospital in Memphis, Tennessee, he joined the Royal Adelaide Hospital Cancer Centre as a consultant medical oncologist in 1998. Professor Brown now works at the Experimental Therapeutics Laboratory in the Hansen Institute at the Royal Adelaide Hospital, and we invite him to talk on what is a clinical trial and why participate. Welcome, Professor Brown.
Thank you very much, Will, for the invitation to speak, and good morning, everyone. What I'm going to talk about is fairly general in overview. I actually didn't plan to talk about melanoma trials that are currently happening other than obliquely, but I'm very happy to talk about that in the questions. So I'll just overview what I will talk about, which is why they are important. I'll give you a bigger context for clinical trials, give you some idea of what they are, tell you what are good trials and what you need to know about joining a trial, and then just give you a story that hit the press a couple of years ago that I think illustrates the complexity of the issues whirling around clinical trials and maybe why a lot of people don't participate, and then really enjoin you to participate. So the two really principal reasons for why they're important is that most cancer treatments today have come about because of trials. This is how drugs get approved. This is the way they do it. And people with cancer are living longer as a result of trials, and Grant's just talked to you about that already. So that's, to me, the primary drivers for patients to consider participating in trials. But on the whole, a lot of people don't take part. And it seems that while this has happened in melanoma, a lot of people have taken part in the recent trials because of the opportunity from them. This may not continue, particularly when the drugs Grant's mentioned, ipilimumab and vemurafenib, become PBS reimbursed, we hope. It could well be that the opportunity will lessen to go on to trials, or at least there'll be fewer patients available if they're getting standard treatment. But the more people who do take part in the trials, the faster we can find better ways to treat and prevent the disease, because at the moment it's not a cure, really, even though there may be long-term survivors with advanced disease. Why don't patients really participate as much as we would like them to? And that's because they often don't know about them. So this exercise is important. They don't have access to them, may live too far away, for example. Some people are not too happy about the idea of being experimented on. There may be issues about affording to participate, probably less in our system, but still an issue. If people have got to travel, they may not actually get reimbursement for some of the visits. And they may have a quite close relationship with the doctor looking after them, and they may not sometimes do it because the doctor doesn't want them to do it. On the other hand, doctors themselves can be a block to participation in clinical trials. They themselves mightn't know about them. They may not want to pass on someone's care to another doctor who is a trials doctor and never see that patient again. They may believe that the treatment they've already got is good enough, and they could be concerned that they'd have a lot more work to do, which would slow down the rate at which they could see new patients. So, as you may get an idea already, this is potentially a combustible mix in which you've got a lot of factors at play. And drug companies make drugs to sell, and there's a process to get them approved to sell. And clinical trials are a means for this happening. And just to give you some indication, it's a very expensive business. There can be perhaps a cost of 500 upwards to $1 billion in the development of a drug for a large market. And that drug might fail, and it might fail in any of the phases of development, quite early or quite late. And you can see for cancer drugs here that failure rate is higher than for drugs in general. And that means that the company that's invested in that has lost money. And they have to recoup that money because they have to give a return to their investors. So this is one of the factors that drives the cost of drugs, and it drives the need for companies to obtain a fair price when they seek approval. Now, I'm not meaning this to be complicated, and I took quite a while to make this and think about it, and I'm not sure that I've quite got it right, but I think you do need to understand that there are a lot of stakeholders here. There are a lot of people who have an interest in getting drugs to market. Obviously, there are the patients, consumers, people who are concerned also about the drug. Many of these people are also voters, so they can have an influence politically. And they often are having someone else pay for the drug. It may be the taxpayer, which could be themselves, or it could be a health insurance company. And that person is, or that entity is going to have a price they will need to pay, which has already been determined in a negotiation with the pharmaceutical company and the regulatory authority. So the central aim, of course, to get a drug is to get a drug approved. And the means by which that happens is the clinical trial, meeting a particular endpoint, a particular outcome, which might be survival, for example. That's the hardest endpoint, and I'll come to this in a bit more detail. And the money that flows to the company allows them to invest, because trials, as I told you, cost a lot of money. And they go through a formal contractual process, sometimes with another company called a contract research organisation, to actually conduct the trial and to run it. And it's a very involved process, and sometimes quite a protracted process. So any drug that's a new drug, and this one that Grant mentioned, vemurafenib, was once in a laboratory, and that's where it's first developed. It has to be shown to work against the cancer in animal models first. And then after that, it might be ready to use in a clinical trial. And a clinical trial is really an experiment, but an experiment with a difference. It's a prospective experiment, so it's one that goes forward. It's not results that you look back on. It is an experiment, so it's not an observation. And it's not a retrospective look back at results where you can actually make what you want of them. Here you're saying what you want to do, you leave that as a fixed question, and you go ahead and answer it. And to do that, you have a document in front of you called a clinical trial protocol. And that really is the blueprint for the conduct of the trial. And that blueprint is something that really is not altered unless there is permission given to alter it. And that permission is given by a research ethics committee that overviews the conduct of a trial. And in that protocol, there's a formula for writing it, but it has to declare what it wants to do, how many people who will participate is predetermined, what sort of people can get into the trial or what aspects they must have to their disease is already predetermined, and exactly how the study will be conducted. So actually, almost day by day, how that will happen. And then what information will be gathered and what your privacy rights are. And then finally, how will the results of the trial be determined and reported. So there are two main phases early on. So the first one is sometimes to just try it out in humans, which may be part of a phase 1 trial, and that's to see whether it causes any side effects. Then as the dose gets increased, you want to find out what is the dose at which it can be tolerated by most patients. And that really then establishes its safety. So is it safe to then use at a dose that's maximally tolerated, then becomes the recommended phase 2 dose. And then that dose is then given to another lot of patients in a separate trial called a phase 2 trial, and that would have more patients in it. And there you're really wanting to see how active that that drug is. And once you've determined that and you've got an active drug, you may then, of course, want to see how it compares with the standard treatment. And that is the phase 3 trial. So the experimental trial usually is put up against a standard treatment, but if there isn't a standard treatment, it might be put up against a dummy pill or a placebo. And those trials, as Grant may have indicated, can run to hundreds of patients. And that trial is a randomised controlled trial, and I'll talk about this in some detail. And that is really the gold standard in clinical research. That provides the evidence for us to make decisions about approval of drugs, for recommending it to patients, for incorporating into guidelines. So the drug, once it's out in the market, of course we continue to learn about it. So there is another phase called phase 4. So once the drug is approved, licensed, you want to obtain data from lots of patients about side effects, particularly ones that would occur less commonly, but you might see in thousands of people. So currently vemurafenib is undergoing a phase 4 trial. It's got about 2,500 patients in the trial so far. So we'll get a lot more useful information about how to use the drug safely. So phase 1 trial, as I mentioned, is to recommend a phase 2 dose, and usually it applies in people who have exhausted all their other treatment options when they've really got nothing left. That is, they've had standard treatments. And they're usually required to have something called a good performance status. And just to give you an illustration of this for melanoma, so Grant had shown you a survival curve earlier. The worse your performance status, the less time you'll have to live. So if you're bedbound largely, then you won't live as long as if you have no symptoms at all. If you have some symptoms and you can't do very much, you'll live not as long as someone who has no symptoms. So this is an important determinant as to whether patients get into a phase 1 trial on the whole. You have to be fit enough to get into phase 1 trials so nothing else carries you off before or confounds any toxic effects the drug might cause. So the usual outcome measure or endpoint for a phase 2 trial is the response rate, and that is something that is measured by a radiologist on a scan. So he looks at the dimensions of the tumour deposits throughout the body, adds them up, and then if it shrinks by more than 30%, that's a partial response. If all measurable disease disappears, that's a complete response. If it neither shrinks or progresses, that is grow more than 20% in dimensions, then that is stable disease. And all of those together, partial response, a complete response and stable disease rate, form the disease control rate. So it gives you some idea of how effective a drug is. And vemurafenib, for example, it's about an 85% disease control rate. So as I mentioned earlier, this randomised controlled trial is the gold standard, and it is used to determine whether an experimental treatment is good as or better than the standard treatment. And you have a control group. So this control group of patients are ones who don't get the intervention, the surgery or the drug, and it enables a fair comparison to be made, a balanced comparison. So randomised controlled trials are really the highest level of evidence we have. And they're actually the basis of something that I guess has been known about for a long time, but has been described more recently as evidence-based medicine. So that really is what should be used to guide clinical practice. May not determine clinical practice, but it would be used to guide it. And it then also becomes the evidence that is used for the regulatory authority to approve a drug, but also for the payer, the health insurance company or the government, to decide whether they will put money behind the drug. So as I mentioned to you earlier, there are a lot of competing interests, and you've got patients, of course, for whom the central question is, will I live longer if I have this drug? You've got the payers who want to know if they'll get value for the drug. You've got patients concerned they might have no choice and they get stuck with the dummy pill, even though they may not know that in some cases. And is there something else on offer? So there are a number of features of a clinical trial that need to be present for it to be considered a good quality trial. And one is that the trial actually has a primary objective. So you must have a reason for doing it. And that becomes the central research question. So that must be ideally a single question, clear and answerable. You must have the power of the numbers of patients in the trial to be able to answer the question. And that question might be to determine if drug A causes melanoma patients to live longer than drug B. And as I mentioned to you earlier, if you decide to measure something like that, you've got to decide and put it on paper and leave it fixed on paper and not change it before you start the trial. So you can't go back and say, oh, look, I don't like the answer, I'll change what I wanted to find out. And the primary endpoint then becomes the way you measure that objective. So it's used to decide the overall result of the trial and actually also helps determine through statistics the number of patients you need to answer the primary question. And survival, as I mentioned to you earlier, is considered to be a hard clinical endpoint. You're either alive or you're dead. So it's considered the most credible endpoint. Yet there are other endpoints you might have heard of, such as progression-free survival. Progression-free survival is not as easy to measure as you might think. It actually depends on a radiologist looking at scans or the investigator and deciding that things have got worse. Now, if they do this away from the centre that conducts the trial, if they don't know which arm that the patients were randomised to, it may be a lot easier for them to determine this, and this is called independent review. And then that gets more credibility to it. There's another very important factor in trials, and that is that trials should be conducted only when you're genuinely uncertain about which treatment is better. And that concept, ethical concept, is known as equipoise and equal balance. And you should really only continue a trial while you're still in that state of uncertainty. And you ethically can't have a trial if it includes a treatment that's not as good as the treatment you want to test. And it really also must include a standard treatment arm. You must compare it fairly with something that's already being out there used to treat patients. You've got to decide who it is who will get into the trial, and for that there are a formal set of inclusion and exclusion criteria, and that's so that you don't end up with apples and oranges. You should really only have apples so you can compare apples with apples. But there can be some pluses and minuses of this approach, so that one plus is that you'll increase the chance that patients will be more alike and that other factors aren't going to confound what you'll need to find out. And therefore it'll make it easier to tell if the drug you want to use is actually having a positive effect or not. But if you do that, you won't make it as generalisable or valid for patients outside the trial, because most of those won't be like some of those won't be like the patients who are actually enrolled in the trial. And that means you'll have fewer people who can get into the trial, so you'll reduce recruitment. But on the other hand, the trial won't be as costly because you won't have as many patients you need to find to answer your question, because they'll be more alike. So you don't want apples and oranges. Took me a long time to find that. So randomisation is really one of the key factors that is important to the design of a trial. And I have this little chap flipping a coin, and in fact one of the first trials it was done that way. The coin was flipped, and that's how patients were allocated to one treatment arm or the other. But in fact now it's done through very complicated computer programs like a computer lottery. And patients have no choice, nor does the doctor have any choice about which arm the patient is allocated to. So they can be allocated to the control group, which might be either standard treatment or placebo, or they can be allocated to the experimental group, so the drug of interest. And the reason that's important is you want all patients, as I mentioned before, to be as alike as possible. And that really is the best way, an unbiased way of determining how effective the experimental drug is. And this is important because there are, unfortunately, practitioners out there who will have drugs or vaccines, for example, that for which claims are made, but those claims aren't based on randomised controlled trials. So they're based on observations, obviously, seeing what happens when you give something. Does it have an effect? But this effect could be a false effect because, as I had indicated to you earlier, just as your performance status determines your survival, how far spread your disease also determines your survival. So if you have stage 4 disease and it's just localised to skin or other soft tissues like lymph nodes, you're more likely to live longer than if it had gone to organs such as brain or liver. And intermediate survival will be that in addition to the soft tissues, it's only gone to lung. So if patients are selected to go into this vaccine trial who just have skin and soft tissue disease, these patients will actually do better anyway. And it's very important to know that the drugs that Grant's mentioned, vemurafenib and ipilimumab, in each case enrolled about 50 to 60% of patients who had the worst outlook. So the benefit that applied for those drugs actually applies to the patients with the worst prognosis. Another important feature that increases the quality of a trial and further reduces the risk for bias is blinding. So you might have heard of this term. And that means that the investigator may not know what it is that's being administered. That's a single blind trial. And you may have heard of the double blind trial, so that neither the investigator or the patient knows what they're receiving. So to give you an example of a currently enrolling trial, so if any of you are not aware of this, I would encourage you to consider it if it's relevant. And this is a trial called the COMBI-D trial. So this trial is looking at its primary endpoint at progression-free survival, but no crossover is permitted, and I think it'll become apparent to you why. So this R step is the randomisation step. So the flip of the coin, if you like. So patients can either get dabrafenib, the vemurafenib-like drug, and another drug that blocks further downstream of a BRAF inhibitor called trametinib. And the other arm has just dabrafenib alone, but it includes a dummy pill. And neither patients nor investigators know which arm the patient will be allocated to. So in effect, it's not really possible to cross over. If you don't know what you're getting, it's not possible to switch over. And there may be toxicity if you try to break the blind and switch over to something that's not going to be of benefit to you. So, as I mentioned to you, you have to have adequate statistical power. You need to have enough patients in a trial, and that is one of the important determinants of whether the question will actually be answered or not. So, patients' rights being protected, of course, is of greatest concern to investigators, and you may be aware of the Nuremberg Code because of the experiments Nazis had conducted during the last war. I think we can still call it the last war, even though there have been a couple since. This code has now been broadened more extensively into something called the Principles of Helsinki, and these define how clinical trials are done. And one of the first steps is, as I mentioned earlier, Research Ethics Committee looks at the trial. And this Research Ethics Committee will have a whole range of people on it. Might include ministers of religion, lawyers, lay people, scientists, doctors, and it often may happen at the hospital where the trial is conducted, and increasingly may happen more broadly even at a state level. And then once the trial is underway, the other very important independent committee, and the Research Ethics Committee is independent of the sponsor, of the patients, of the investigators. The other independent committee is the Data and Safety Monitoring Board. And so this has a very important role of ensuring that risks to patients are minimised as the trial is conducted. It ensures that there is no tampering of data, and ensures that a trial will be stopped if there are concerns about safety, or if the objectives of the trial have been met, that is, to make sure the trial doesn't go on too long, and either deny patients access to a good drug. So I will explore this in a bit more detail with this story that I was going to tell you about. So an example here is this BRIM3 trial, which is the randomised controlled trial that led to the approval of vemurafenib. And in this trial, there were 675 patients who were randomised to receive vemurafenib, a pill, or dacarbazine, an injection. So there was no way that this could be a double-blind or even a single-blind trial. Everyone knew what they were going to get. And there was no crossover permitted. And the US Food and Drug Administration, which is the American equivalent of the Therapeutic Goods Administration here, had allowed the company to market the drug, would allow a company to market a drug based on an early promise, because there already clear evidence that this was an active drug in patients whose melanoma had the BRAF mutation, as long as they proceeded with a randomised controlled trial to compare it to standard treatment. But there's a sting in the tail if they did that, because if they went to market the drug while they're getting the trial organised, then it'd be very hard to enrol patients to that trial, because half of patients would be randomised to receive the control drug, the dacarbazine, which is much less effective for patients with BRAF mutant melanoma. But this is the way to ensure regulatory approval, at least as it is currently construed. So there are also concerns by the investigators in this trial that they needed to find out whether patients would survive longer as a result of having this drug. And these concerns, which Grant kind of alluded to, you may have noticed when he showed that ERK staining in the tumours, that perhaps there would be new cancers that would be kicked off by using this drug. And in fact, there are new skin cancers that are kicked off by using the drug, but they can be safely excised. But there's still a residual concern that internal cancers might be kicked off by this drug. And if they did happen, obviously that would be a disaster in someone with advanced melanoma. It might actually cause them to live a shorter time, irrespective of whether they benefited initially from the vemurafenib. And the other problem that confronted the investigators, because they had none of the knowledge we have now, is that once you stopped the drug because the disease had got worse and the drug was no longer effective, that maybe the disease would take off, maybe grow even more viciously than it had before. And that concern, I think, has now been put to rest by an analysis of this BRIM3 trial. But these were the concerns facing the company and the investigators at the time this trial was planned. And originally, the overall survival was the single primary endpoint. But once the drug was known to be active, and after further discussions with the FDA, the overall survival and progression-free survival together became the endpoints of this trial. And there was a plan, so there's always a statistical plan set up for a trial. And that plan, if you're looking at overall survival, will depend on a certain number of deaths being accrued. It doesn't matter whether those deaths have occurred in the control arm or in the experimental arm. And there was a plan that at 50% of that projected number of deaths, which is 196, so 98 deaths, there would be an interim analysis for overall survival, so that the independent Data and Safety Monitoring Board would review those data. And that's indeed what happened. And what they found, and this was published in the New England Journal of Medicine, what they found is the investigational drug, excuse me, vemurafenib, did reduce the risk of death by 63% compared to dacarbazine. So it was a beneficial drug. And at that point, patients were allowed to cross over from dacarbazine to vemurafenib. In contrast, this other drug, dabrafenib, which we've mentioned already, is a drug that has been developed after vemurafenib, so some of the information about the activity of vemurafenib has been available. And you can see here the design of this trial is quite different. So you have similarly large number of patients being enrolled, in fact some more, and this time the primary endpoint is not overall survival but progression-free survival, and patients are actually randomised to just dabrafenib alone or to dacarbazine, DTIC, the same drug as in the vemurafenib or BRIM3 study. But the randomisation is unequal, so the dice is loaded in favour of the patient, if you like, by having for every 4 patients, 3 will go on dabrafenib and 1 will go on dacarbazine. And the other thing that's been permitted is that patients are allowed to cross over when evidence on their scan shows that the cancer is getting worse. So the patients who have been on dacarbazine, where they're more likely to progress, will still have the opportunity to receive the drug that's now believed to be better because it works very similarly to vemurafenib. So the story I want to tell you about is this quite dramatic story in the New York Times, which was published a couple of years ago at the time the BRIM3 trial was being undertaken. And they're 2 first cousins who actually used to play together. And one called Brandon, who's actually, I think, on the right, and Tommy, who's the one on the left. And this is extracted from the article. So Brandon wasn't... So what happened is that in the trial, Brandon got allocated to the dacarbazine arm and Tommy got the vemurafenib arm. And anyone who knows anything about vemurafenib will clearly see this patient is on vemurafenib because he's wearing this extraordinary headgear and a whole lot of stuff because of the photosensitivity you can get from vemurafenib. And the views of each of these 2 patients was quite interesting. And Brandon felt that Tommy should be the one getting the treatment. This was the right thing because he actually had a couple of kids. And but actually Tommy thought his cousin should get it. And in fact, he got quite concerned about the way this was being run, that his cousin wouldn't be able to get access to this drug at all, and got quite upset about it. And you can see this is a real ethical dilemma. And the leader of this trial, Dr. Chapman at Memorial Sloan Kettering Cancer Centre in New York, believed that it was actually very important to determine whether patients would truly benefit for all the reasons I mentioned to you earlier. And the doctor who was actually caring for the 2 patients was in a particular quandary because he had to explain to the mother of one of them why he wasn't getting vemurafenib. But he still complied with the trial requirements that the randomisation as allocated proceed. But there was clearly disquiet in the medical and the scientific community as reflected here. And I won't read the whole thing, but I'm not sure how many people can actually read it. But there's some concern by a prominent clinician scientist that drugs that have minimal side effects and dramatic response rates, and where we understand what's going on, why do you have to be so rigorous about the way these trials are conducted? Even the person responsible in the FDA for this area did make the comment that because this drug was so effective compared with a drug that was relatively ineffective, that this is an unprecedented situation that will hopefully be increasingly common. And it may require regulatory flexibility and an open public discussion. Said like a true bureaucrat, I thought. So it really is a genuine dilemma with no easy answers. And Dr. Chapman, reiterated this point that it's good to see tumours disappear, but that's not really what it's about. What it's about is ensuring patients will live longer. On the other hand, another clinician scientist felt he would see it very difficult to just accrue the number of events required, that is the number of deaths required, by allowing people on the control arm to die without any further intervention. And some people felt that even this concept of equipoise was being breached, that's an ethical code that says if doctors need to be genuinely uncertain about the benefits of the drugs that are being tested. And here there seemed to be some evidence that one drug was more effective than the other. But Dr. Chapman really wanted to find out if the drug was going to work. He said, if we never know that, we'll never be able to build on anything. And I mean, now with a retrospective scope, I think it's become clearer that despite the grief, that is the right decision that's been made, as reflected in the last comment. So what to expect when considering a clinical trial? So the clinician investigator who talks to you should really give a full and frank discussion of the treatment options, and those discussions should include the standard treatment, and they should also include the no treatment option. They should discuss why the research is being done and any potential risks and benefits. Patients should be allowed to have time to review the options in consultation with family and friends. There shouldn't be any rush to sign up. And they need to also understand what's called the assessment schedule. So that is how often they need to come to the hospital, when they get tests, and how the treatment's given and how often, and what the convenience will be. So the benefits of participating in a trial are going to be that at a minimum, the patient will receive the best standard treatment. And that's this question of equipoise. And if the new treatment or intervention is proven to work, then you may actually be benefiting, at least have a certain percentage chance of benefiting. And patients, of course, if they're altruistically motivated, will see they could help others and improve cancer care in general. But there are risks to any trial, and these risks include things we don't even know about. So trametinib, this drug I mentioned, for example, can cause, rarely, a blockage in a vein behind the eye. And as a class in general, which is a MEK inhibitor, people have lost vision and not recovered vision. So this was really quite unexpected. And the new treatments may in fact not be better than standard care. And even if it had benefits, you personally may not benefit. So you always have to realise that going into a trial, you personally may not benefit. And that's something that's often discussed in the informed consent process. And once you're given that informed consent, that really marks the point at which you are enrolled in the trial. And then you'll be randomised. And that point of randomisation is often taken as the time from which the counting occurs, the number of days your involvement in the trial is. And it also marks permission for the study-related procedures, the extra tests to be done that might even determine whether you actually stay in the trial. And that period in which that assessment is being made is called a screening period. And that, as I said, could determine ultimately your eligibility for the trial. And there's an obligation on patients that if they are enrolled in the trial, they need to stick with it. They can't go off and do things that are not prescribed in the trial, or proscribed, I should say, in the trial. Of course, if you don't want to do that, you're free to opt out of the trial any old time and get any treatment you want without fear or prejudice. So sometimes patients are interested in trials because they know current therapy is not good enough. Even though there's uncertainties about new treatment, they may be having a lot of trouble with the current therapy. They may want to help other patients. They may want to help the doctor involved. They're likely to get improved care. It may affect favourably the outcome of their disease. There's potential, obviously, for access to new treatments that are not approved, that aren't on the market. And patients will generally get a really thorough going over. Anyone who's been in a trial will know that the doctor spends a lot more time with you than you otherwise have spent with you. and that there's more opportunity usually for discussion, and that you get a lot of information on your condition. And you get a lot of focus on your problems, and you get a lot of contact with experienced people, and you can actually plot out what your moves might be for future care and treatment. So that really is the end of my talk, and I'll just show you here a couple of websites, including our own NHMRC website, where you can gather further information if you wish.